Okay, hello everybody and welcome to this little lecture from the University of Aberdeen looking at making medicines and the drug discovery, development and regulatory process. What I hope to do today is just give you a summary and a high-level overview of how these things work. So how this event's going to work is it's going to be a question and answer session, which I would ask you to use the Q&A section at the end. There's not going to be questions during the presentation, but all the questions will be answered at the end. There isn't a chat function available, but if you want to ask a question, please use the Q&A. Questions will be moderated by my colleagues, Susan and Rianne, and don't worry, they will appear, but they won't necessarily appear right away. Questions can be added to the Q&A at any time during the presentation. So, my name is Heather Wallace and I am a professor here at the university in biochemical pharmacology and toxicology, and I work in medicines development and regulation. So let's think about what medicines have done for us over the years. So here you can see a slide that looks at what's happened perhaps in the last 100 years. Our life expectancy has increased or doubled by a factor, increased by a factor of 2, or indeed doubled. Infant mortality has decreased by a factor of 5. We can now treat chronic diseases such as diabetes, cancer, asthma. We've cured infectious diseases, although coronavirus is obviously still an issue, and we have prevented diseases such as smallpox and polio. So, medicines have really helped and improved our quality of life. And when we look at medicines, there are medicines and there are therapies. So what you can see on this slide are the classic small molecule drugs that you can see here. We also have antibody drugs. Now, these are more complicated drugs that are protein-based, and they are often related to cancer therapies. We have peptide drugs, again, more complicated, and we also have vaccines. So these are the kind of agents that we would classify under medicines. And over here, I have what we would classify as therapies. So here we have gene therapy, where you replace a faulty gene with a correct one. And then over here on the far right, we have cell therapy, where you target cells to, for example, kill cancers. What I'd like to do today, however, is talk to you about small molecule drugs and how these have helped improve our quality of life over the last 100 or so years. So when we look at medicines, what really matters in terms of medicines? Well, the most important 3 criteria are shown on this slide. So efficacy, the drug must do what it says on the tin. So it must be effective in either treating the symptoms of a disease, curing a disease, or preventing a disease. The medicine has to be safe. So in other words, there has to be very limited toxicity, few side effects. All drugs, all medicines will have some side effects, but the aim is to have the minimum number of side effects in all medicines. And finally, the quality, we need to have a high standard of manufacturing, a reproducible quality, and that is absolutely vital for all medicines that are approved. Now, the people that look into these things, the people involved in efficacy, safety and quality are quite different. What you can see here is the efficacy is usually clinicians, clinical pharmacologists and research nurses and research staff. The safety would be toxicologists and pharmacologists in the main, but also some clinical scientists and pharmacists. And the quality would be the pharmacists, the engineers, and those people involved in the manufacturing process. So a huge number of people involved in the overall medicines process. What I want to do now is look at the process of drug discovery and development. This is how we make medicines. And the first thing I want to point out is that medicines, drug discovery and development are actually 2 processes. This first part is the drug discovery event, and this second part is the drug development part. And then this part in orange is the regulatory process, and the part in blue is when we look at what happens when the medicine is out in the community and millions of people are taking the drug. The point of this latter part here is to look for idiosyncratic side effects that might not be possible to detect in a clinical trial where we're maybe only looking at 10,000 people. It's where we see effects that occur when it's 1 in a million or 1 in 2 million people. So the drug discovery, as you can see here, is the basic research. It's medicinal chemistry, it's biology, and then the preclinical toxicology and pharmacology. The development part is the clinical development, and there are 3 phases to clinical development. Phase 1, which is human volunteer studies, phase 2, which may involve volunteers and patients, and then phase 3, where we look at patient studies and do comparator studies. Now, once all these processes have been completed, you can then submit your dossier to the regulatory authorities, and with any luck, you will get your medicine approved. Again, large numbers of people involved. You can see here the three parts. The drug discovery, it's medicinal chemists, computational biologists and biologists. The development is toxicologists and pharmacologists again, with some clinical scientists, pharmacists and clinicians. And then the approval process is the regulators, and that's made up of the national competent authorities. And you can see here the US National Competent Authority, which is the Food and Drug Administration, and the UK National Competent Authority, which is the Medicines and Healthcare Products Regulatory Agency. So different people are involved at different times in this drug discovery and development process. So I want to show you now the model of this drug discovery, development and approval. And this is the classic drug discovery funnel, we call it. And what you can see here at the far left is the starting process, the drug discovery. Now this might have, it says here 15,000, but it might have 100,000 or 1 million compounds to start with. These are then looked at and assessed, and some, much fewer number, go forward to the preclinical. From the preclinical, the successful ones will go forward to the clinical trials. These are much more expensive than the preclinical work, and so fewer numbers go forward. And from this clinical trial, you may get 1 successful medicine. Now, there are very important points in this process. The first is that it costs a lot of money. At the moment, it's estimated that $2 billion to develop 1 single drug. The other problem is that it's time consuming, very time expensive, and you can see it can take 10 to 12 years for this process here to be successful. Now that's a slight problem because all this process, this drug discovery and development process, is covered by what we call a patent. So a company would patent its drug somewhere around the discovery phase, and once the patent was granted, it would have 20 years of a license or a right, or not a license, it's a right to develop that medicine to the point of approval. And no one else, no other company can interfere with that development at that time. So that means that you have a total of 20 years when your company has exclusive rights to your potential medicine. Now, the problem is that you spend money from here all the way to here, and you only make money once you have your drug approved. And that's why it's a very expensive business. Now, on this slide, I'm trying to show you what the process is. So you have an idea, you have a target, so that's an idea for a new medicine, you have a disease that's got an unmet medical need, and basically you spend until you get to this process where, if you're lucky, you will get a marketing authorization from one of these national competent authorities for a drug. You then have a medicine, and you can sell that medicine, and then you can make money until your patent expires after your 20 years and basically generic competition comes in. So this is the pharmaceutical company business model, this whole process. And this model is one where only money is made in these sort of 8 years here, but you have to cover the costs not only of this successful medicine, but you have to cover the costs of all of the failure of the medicines that you didn't develop. So the reason medicine development is so expensive is because we start with so many compounds and we have so few successes. So the attrition rate is something that the pharmaceutical industry is really concerned about and is working very hard to improve that attrition rate. So let me now move on and think about why medicines are regulated. So why do we regulate the use of medicines? Well, the reason is that in the 1950s, late 1950s and early 1960s, a medicine was used called thalidomide, which caused major phocomelia, major problems in unborn children, and that led to what we knew then as the Medicines Act of 1968, which basically said that medicines had to be tested and tried before they could be used in individuals. We now have a new Medicines Act, the Human Regulation of Medicines Act 2012, and that is basically an act that says that you have to have a licensing system, and that licensing system provides a marketing authorization. And the 3 essential criteria for getting a marketing authorization are that you have a safe medicine that is effective against the target and the disease that you are studying, and that the medicine manufacture is of high quality. So that's the regulatory process, if you like. And regulation, as you can see here, involves the regulatory authorities. It's an approval process, and it's national and international. And I'll show you a little bit about the national authorities later on. What that approval process includes is all the features of the medicine. So it's the physical chemical properties of the medicine, whether it's a tablet or whether it's an infusion or a solution. It's the pharmacological properties, making sure that it does what it says on the tin, it attacks the right target. It looks at the safety of the medicine, so that's the potential for adverse effects, the potential for toxicity. It covers the clinical trials, so they all have to be positive. And it also covers what I mentioned as the post-marketing surveillance or the pharmacovigilance. So all of these things are included in the regulatory process. So the regulators, who are they? Well, they are what we call national competent authorities. These are authorities given the permission by governments to look into medicines, medical devices, etc., and to have the power to approve or not these medicines or devices. The main ones are the Food and Drug Administration, the FDA in the United States, the Medicines and Healthcare Regulatory Products Agency, or the MHRA in the UK, and the main committee for that is the Committee on Human Medicines, the CHM, and then the European Medicines Agency, or the EMA in Europe. So these are the ones that would really affect those of us who are involved in medicines regulation in the UK. So here's the MHRA, the Medicines and Healthcare Products Regulatory Agency. It is a combination of the Medicines Control Agency, which was medicines, and the Medical Devices Agency, which looked at implants and things. And basically it's a government agency based around Canary Wharf in London, and its remit is to ensure the safety and appropriateness of medicines, and that's both medicines and medical devices. And as I've said before, medicines, there's no product that is free of any risk. So the idea or the main purpose of the MHRA and the license and the marketing authorization is to ensure that the risks to individuals are minimised. So medicines are approved and they're given this marketing authorization. Medical devices are approved, and that includes things like silicone implants, new knees, new hearts, heart valves, hip joints, knee joints, wrist joints, anything that is effectively considered a device. And the MHRA is as I say, a regulatory body, but it's also a body that is involved in developing future policy for medicines. So it works with the government to advise ministers on safety and future policy. It looks to the public and it helps with information gathering, and there is a lot of information available on the MHRA website which will help with, for example, they do something called a drug safety update where they write reports on current positions of new medicines and indeed old medicines and any safety alerts that have been brought forward. The MHRA is also accountable, and you can see here that they can withdraw a product from the market if there are what we call safety alerts. They can suspend production if, for example, a company changes the manufacture of one of the components of a medicine. And they can indeed prosecute if someone breaches any of their regulations. So it's the MHRA is not just an advisory body, it actually has powers to prosecute and suspend the use of medicines. So the agency is committed to developing regulation that is accountable, proportionate, targeted, consistent and transparent. And these are the 5 key features of regulatory process. So how does the regulatory process work? How does the MHRA work? Well, this is a very simplified diagram of what happens. Basically, it is a committee structure. Here you can see on the right-hand side, you can see here the committee, which is the main committee, which is called the Committee on Human Medicines, or the CHM. And that's the committee that makes decisions. But this committee has beneath it something like 17 or 18 expert advisory groups. And these expert advisory groups consist of invited experts, MHRA scientific officers, and these expert advisory groups can be, for example, on pharmacovigilance, paediatric medicines, oncology, neuropharmacology, pharmacy, any number of different expert advisory groups. And I have been on the Paediatric Medicines Expert Advisory Group since 2009. So I'm part of this process that then produces advice, which then goes back to the CHM, which then makes decisions. So what the EAG do, as I say, is review information which is provided by the MHRA scientific officers. These are dossiers, usually a couple of 100 pages long. We read through these and advise, and that advice is then fed up through the system. Only people who can approve yes or no are the Committee on Human Medicines. So I hope that has given you a very, very quick overview of the process of making medicines and how they're regulated. What I've done is I've indicated that the medicines and therapies that we have and have developed over the last 50, 60 years really have improved quality of life. And there are many forms of these medicines and therapies. For example, there are drugs, small molecule drugs that we've talked about that manufactured today. There are vaccines, there are cell and gene therapies, there are antibodies, and there are peptide drugs. I've overviewed the drug discovery and development process. I've explained how it works and the expense and the time-consuming nature of it, and why sometimes medicines are so expensive. I've summarised the regulatory process and explained the 3 key criteria, which are efficacy, safety, and quality. So I hope that you have enjoyed that little lecture here from the University of Aberdeen, and I'm very happy now to answer any questions if anybody would like to ask them. Thank you. Thank you for your attention. Thank you for listening.